So what are the ideas that have been behind the improvements that Matt showed a little while ago? Now, these are my disclosures, as usual. Everyone's accompanied here, I hope. So, when I was a trainee registrar at Prince Alfred Hospital in the 1970s, I remember very well one day, Tom Wilkinson, one of my bosses, came up to me with that month's issue of Blood. And he opened it up and showed me this paper here, and he said, "Look at these crazy bastards, look at what they're doing. Are they completely mad?" And of course I had to say yes, because he was the boss. But this is one of the foundation papers of marrow transplantation, clinical transplantation from Seattle. And it describes, as you can read there, 100 consecutive patients with acute leukemia who were treated in that centre in the early 1970s, and summarises what happened to them. And as you can see in the highlighted bit in yellow there on the left-hand side, out of 100 patients, 13 patients are alive with a marrow graft on no maintenance anti-leukemia therapy 1 to 4.5 years after transplantation. So I think this is the reason that there was a bit of scepticism in the mid-1970s about the value of marrow transplantation. There was also some scepticism about the final sentence there on the right-hand side, this observation coupled with the observation that some patients may be cured of this. It indicates that marrow transplantation should now be undertaken earlier in the management of patients with acute leukemia who have a matched sibling donor. And some people thought, wow, 13 out of 100, is that really worthwhile? However, my boss and I were obviously wrong, because only 13 years later, Don Thomas, who was the lead author on that paper, got the Nobel Prize. So he must have been doing something right. So Dr. Thomas was honoured for endeavours in experimental and clinical bone marrow transplantation. Their prizes emphasise the importance of patient-related research. I remember the first time I met Don Thomas was in 1985, some years after this, when Sydney hosted the International Haematology Society meeting. They had an international society in those days before America took over. And we made the misjudgment of inviting Richard O'Reilly from New York to give the plenary talk on bone marrow transplantation. And the day before, we found out that he wasn't going to show up. So we were left without a speaker. So I remember Jeff Scher and I frantically tracked down Don Thomas, who happened to come to that meeting, and we got down on our knees and said, "Dr. Thomas, will you give a talk tomorrow in place of our invited speaker?" And we thought that was a big ask, but he said, "Oh, I think I'll be able to do something about that." So sure enough, the next day he arrived with a carousel full of 35mm slides and gave an incredible 1-hour talk on the current status of marrow transplantation in 1985, and that's the sort of guy that he was. It was also the cheapest international speaker that we got for that particular conference. So this is just a random image that I culled out of the Australian Transplant Registry just to illustrate what Matt just showed before, that between 1977 and whenever it was, this is 10 years ago, there was a distinct improvement in the overall results for marrow transplantation, such that, as you can see in that blue curve there, more than half of patients in this particular category were actually long-term survivors of a transplant. So that's a big difference over 2 decades. So there are 10 concepts that I picked out as being worthy of further thought. And I'm going to discuss all of these, believe it or not. So these are the ideas that really underpin the field. So what I was interested in was, where did these ideas come from? So I actually went back and pulled out the literature on these concepts and I'm going to discuss these. So the first one, or the first two that I've rolled together, are that hematopoietic cells is a source of blood cell production, and this can be used for rescue from bone marrow damage or aplasia. So where does this come from? So the original publication on this dates back nearly 70 years. So this is a paper in the Journal of National Cancer Institute in 1950 by Lorenz, Hoff, and a few others from Bethesda. So this describes, in very briefly, giving bone marrow cells to mice and guinea pigs that have been given lethal doses of irradiation. And this is a very classical image. It's one of the data images in this paper, showing, it essentially sums up all we know about marrow transplantation now. Just see if there's a pointer here. Sorry? Oh, anyway, doesn't matter. Yeah. So we can see that irradiated guinea pigs given marrow cells survive this experiment, but the ones that weren't given anything all die from the consequences of aplasia. So in a sense, because these are inbred guinea pigs, this is a syngeneic transplant from an identical twin, as it were, a genetically identical donor. And it shows recovery of bone marrow function after this transplant. And the conclusion from this paper is it's tempting to assume that the action of protection by bone marrow injection consists of providing new areas of hematopoiesis. And this is contrary to an assumption of a paper published 2 years before, where similar experiments were done, except that the spleen was shielded from the effects of radiation. And the authors, the first author was Jacobson, postulated this was due to a humoral rather than a cellular factor. And the cellular nature of the protection was confirmed in 2 papers in 1956. This one he published in Cancer Research, the first author being Peter Knoll. And there'll be some people in the audience who will recognise the name Knoll as part of Knoll and Hungerford, because Knoll subsequently moved from San Francisco to Philadelphia, to the University of Pennsylvania, and described about a decade later on a unique chromosome in chronic myeloid leukemia, which they called the Philadelphia chromosome. So he had a fairly illustrious career. You'll also notice that the institution that Knoll worked at the time was the US Naval Radiological Defense Laboratory. So in the 1950s, much of the research on bone marrow transplantation was actually done in various countries by government-funded organisations interested in the effects of nuclear energy for some reason. And the conclusion from this paper is that injected rat marrow cells survive, divide, and eventually repopulate the marrow cavity of irradiated mice, and prolonged survival, in some cases beyond 30 days, appears correlated with the growth of phosphatase-positive marrow. That's rat marrow. So that the rat marrow is actually functioning hematopoietically in the heterologous host. This seems fairly self-evident now, but it wasn't in those days. As an immediate corollary to what I've just said about hematopoiesis being transferred, it was recognised at the same time that the immune system was also reconstituted by the transfer of hematopoietic cells. So this was published in the same year by a group from the Atomic Energy Research Establishment in Berkshire in the UK. And in this paper they describe using a particular mouse strain that had a marker chromosome, that they're able to see the marker chromosomes which belong to the donor, not only in the marrow of the reconstituted animals, but also in the thymus lymph nodes and spleen, which implied that immune cells were also being involved in the repopulation. And as another consequence of that, it was recognised at around about the same time that if you did this, and then there was a genetic disparity between the donor and the recipient, that the animals that survived subsequently developed a strange wasting disease, which was initially called secondary disease, but then later on called the graft versus host reaction. So this was first described, as far as I can find out, in 1957 by our old friend Delta Uphoff from the National Cancer Institute, where this graft versus host reaction was defined. Another person who was working in this field at the same time, again from a National Defence Research Laboratory, was Dirk van Beckum. So I met Dirk van Beckum in 1978 at a very early European bone marrow transplant group meeting. He was then at the end of his career, but he was still a very formidable individual. So this is pathogenesis of secondary disease after foreign bone marrow transplantation in irradiated mice. And the conclusion here is that evidence has been provided in support of the hypothesis that secondary disease in mice is treated with foreign bone marrow is due to a combination of late radiation damage, an immune reaction of the graft against the host, and impaired immune defence against microorganisms. So in this paper, there's a beautiful hand-drawn diagram, which I couldn't reproduce, which basically shows what he feels is the interplay between these factors. Very insightful. About a decade later, it was definitively proven that this was actually a reaction that was due to genetic disparity rather than inflammatory change, because the same sorts of patterns were found in germ-free mice, which had no inflammatory reaction. Then there were people who decided that something had to be done about the graft versus host reaction, and could that be modified to allow successful transplantation in outbred animals where there are inherent genetic disparities despite matching for MHC. So the Seattle group was predominant in this, and this paper, which is the earliest one that I can find in outbred animals like dogs and people, comes from Don Thomas. So this was published in Blood in 1962, and this work was carried out by Don Thomas in Cooperstown, Massachusetts, before he moved to Seattle. And amongst other things, this shows the whole basis of successful marrow transplantation in MHC-matched individuals, outbred individuals. So this is a litter of 4 dogs, and one of the dogs is the donor, and they didn't know anything about tissue matching at that time, so this was just guesswork. But you can see that one dog, the one at the top there, 392, was a survivor. It lived long enough to be able to survive the effects of the radiation, and it reconstituted its white blood cell count, indicating that the donor marrow was functioning, whereas the other 2 dogs did not survive. They rejected their grafts. And although they didn't know at the time, clearly dog 392 was DLA matched with the donor, and the other 2 dogs were mismatched. The other thing that this shows is that this group also tried to modify the graft versus host reaction by giving an immunosuppressant drug, in this case methotrexate, although cyclophosphamide was also trialled at the time. And this was successful in reducing the graft versus host reaction in these dogs. Subsequently, this was modified, and this is from Don Thomas's group when he moved to Seattle in the 1980s, exploring further the use of methotrexate regimens for control of graft versus host disease. And these are in DLA mismatched dogs, and it shows that giving a protracted course of methotrexate, which is the solid line there from day 1 up to day 102, that's a proportion of these mismatched dogs could actually be successfully made long-term survivors. In other words, you could control the graft versus host reaction in these animals. The other important drug that was introduced into clinical practice about a decade later than this was cyclosporin. And one of the earliest groups to explore this was the Marsden group in the UK, with the first author of Ray Poles. I think some people in the audience may remember him. And this was then taken up by other groups, and in this case, this is by the Seattle group. And there are people who say that you can't do phase 3 randomised clinical trials in marrow transplantation because it's just too difficult. However, the Seattle group managed to do that with cyclosporin. So this is a paper by the first author, Joachim Dieg, published in Blood in 1985, showing a group of patients with AML receiving GVHD prophylaxis with either methotrexate or cyclosporin. The conclusion of the paper is that there was no difference in the protective effect of the two drugs. These are given as single agents, and you can see the p-value there. However, you notice there's only 39 and 36 patients in each arm because it's hard to do a randomised trial in marrow transplantation. But you do get the impression that cyclosporin probably is better than methotrexate in this particular situation. The next year there was again a publication from the Seattle group showing that the combination of methotrexate and cyclosporin is better than methotrexate alone. Again, small numbers, but there was actually a significant p-value associated with that difference. So this essentially provides the basis for current clinical practice of combining a drug such as cyclosporin and tacrolimus, a calcineurin inhibitor, with a drug such as methotrexate, which is designed to reduce T-cell proliferation. Now, tissue typing. Of course, the mouse immunogeneticists knew about genetic differences between inbred mouse strains back in the 1940s. And this was all very well worked out. And it was understood that you could not get graft versus host disease if you transplanted marrow cells or hematopoietic cells or lymphoid cells from one inbred mouse to one of the same strain, but you could if you cross strains. But this really isn't particularly relevant, again, to outbred organisms like us. So a lot of this stuff was worked out in dogs, again, by the Seattle group. So as you can see here, this was published in 1968. It was found that you could immunise dogs with leukocytes from other dogs and produce anti-sera, which then identified genetic differences between various dogs. And this was broadly applicable. So, as it says here, testing of lymphocytes from 62 unrelated dogs with 4 anti-sera gave positive reactions with those particular frequencies, fairly high frequencies. And then this was then tested amongst siblings, where there were obviously inherent genetic differences that were dictated by the major histocompatibility locus of these animals. So none of the mismatched recipients lived longer than 14 days, and all of the matched recipients lived beyond 40 days, etc., etc. So cross-immunisation of littermates produced reasonably specific typing sera. And they then followed this up fairly promptly by doing transplants between littermates using the information based on tissue typing. So this paper describes, again, some of the essential elements involved in marrow transplantation, that you immunosuppress the dogs with cyclophosphamide, you give marrow cells from a DLA-matched littermate of that dog, and you can produce long-term chimerism in that particular animal. So the basis for tissue typing in humans goes back a long way as well. So this is one of the earliest descriptions back in 1967 by Bach and Amos, stating as here, a single locus with 15 or more alleles controls reactivity in mixed leukocyte cultures. This is when you mix the lymphocytes of individuals together and see whether they react with each other. And at the bottom, it is proposed that this is the major histocompatibility locus in man. And they then followed this up by doing skin grafts between matched siblings and finding that these actually took. That would be something that would be pretty hard to get through the ethics committee these days. And not long after that, in 1970, this paper appeared in The Lancet, and saying that they looked at the tissue typing records of 500 unrelated individuals, and using a computer in 1970, it must have been quite a feat in itself, they found 4 out of the 500 that were phenotypically identical for the HLA locus. And since it is certain that some HLA antigens cannot yet be recognised serologically, they then subjected these cells from these 4 individuals to a mixed lymphocyte culture to distinguish true from pseudo-identity. So 2 of the 4 were actually serologically identical for HLA antigens and showed a lack of stimulation in MLC. And then this finding suggests that given an organ exchange scheme, blah, 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 it might be possible to select, for example, cadaver donors for kidney grafts. However, you could also use this information for selecting marrow donors, unrelated marrow donors, and a whole industry is developed out of this, such that there are, I've forgotten how many, 20, 30 million HLA-typed individuals around the world who are prepared to donate bone marrow or peripheral blood stem cells to worthy individuals that they don't know. So this really is the foundation paper for that whole enterprise. I don't know who read The Hematologist recently, but there was an obituary for the first author of this paper, Johannes van Root, or Jan van Root. I met him in one of these EB&T meetings, which was always held in a small skiing resort in the French or Swiss Alps in the 1970s. And I think it was in 1979. And he was remarkable for 2 things. His incredible brain. He was firing away at everybody, I remember, at that particular meeting. They were always small meetings. And the second thing he was remarkable for was everyone else used to catch the chairlift up the mountain. He didn't. He just skied up the mountain. So he published 550 scientific papers. The last one was a couple of months before he died at the age of 91, and he was the first author on it. Not bad. So the data on tissue typing was then rapidly used in clinical transplantation. And this paper, published by the Seattle group in 1972, shows the results of the first 4 patients with aplastic anemia who were treated with an HLA identical sibling transplant. So there were 4 patients. They were matched siblings that tested for HLA compatibility, and they donated marrow. They were given methotrexate as to modify the graft versus host disease, and they all engrafted. 2 of them died, but 2 patients were long-term survivors. So this is again a foundation paper for the use of marrow transplantation in this particular setting. Now, that's okay for a small subset of people with marrow failure. What about the major application of allogeneic transplantation as it is today, which is to treat malignancy? So the next concept is the recognition of the anti-tumour or anti-cancer effect of homologous or allogeneic transplantation. And that's now known as the graft versus leukemia reaction. Where did this concept come from? There was a lot of work in the 1940s and early 1950s in mice and other experimental animals looking at the possibility of harnessing the immune response to cure cancer. But the first paper that I can find, and again it's fairly widely cited, using marrow cells to do this, comes from this paper in 1956 published in the British Medical Journal by Barnes et al. Treatment of murine leukemia with X-rays and homologous bone marrow. This is a very curious paper because it's very short, very not much detail in it, and it implies that homologous bone marrow does something. But actually, when you look at their conclusions, they say that blah, 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 the results would indicate that for this purpose, isologous, in other words, autologous marrow taken from a donor of the CBA strain, because there were CBA recipients, would be superior to homologous bone marrow from strain A. So why is this the foundation paper? I had to actually read the data very carefully to understand why, and they don't actually make this point. But you can see that, sorry, this is a bit confusing. Just ignore 1, 2, and 3 at the top. They gave all of the mice leukemia cells, the unirradiated controls all died of leukemia because there was nothing to hold it back. If they gave mice a huge dose of radiation, 1,500 rads, which is clearly near the lethal dose for the leukemia, then a lot of the animals survived, although a few actually died of progressive leukemia, showing that it was a threshold dose. These mice here are the ones that are mismatched from different strains. And you can see over in the third column at the bottom there that some were cured, and we don't know whether that was because of radiation, but quite a few died. But the point that I picked up from this paper is that the mice that died actually died of graft versus host disease rather than of leukemia. So there weren't actually any leukemic deaths in this. Anyway, that be as it may. This is a fascinating paper. Everyone should read this. So this was done by Don Thomas when he was in Cooperstown, in New York, and this paper is incredible. So this is human work. They gave bone marrow to patients with cancer, and the idea was that if you give a huge dose of alkylating agent or radiation, that you may be able to cure the leukemia in patients with advanced disease, and then rescue them from the effects of that on normal marrow function by giving a transfusion of bone marrow cells. So there are actually 5 case vignettes in this paper. They didn't know anything about tissue typing. They didn't know anything about the dose of radiation needed to do this. It was all experimentation. And the 5 patients had end-stage CML, CLL, myeloma, lung cancer, ovarian cancer, and they gave marrow from fetuses, where they scraped out marrow cells from fetuses, or from cadavers, recently dead people. There's no mention of this being cleared by the ethics committee or any considerations like that. You could apply a whole range of adjectives to describe this. I suppose courageous would be one, heroic, foolhardy, but essentially this is very early stuff. And the incredible thing is that these people kept on doing this. Amazing. And I just love this bit in the discussion. If you go halfway down, in an atomic age with reactor accidents, not to mention stupidities with bombs, someone is going to get more radiation than is good for him. This is in the New England Journal of Medicine, believe it or not. If infusion of marrow can do blah, 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 the leukemic patient who needs radiation and bone marrow, or the uremic patient needs, are the people who deserve immediate consideration. From helping them, one will be preparing for the atomic disaster of tomorrow, and not the other way around. So that's very interesting, the perspectives. And then, of course, after, from 1957 to 1977, a lot of water went under the bridge. So in man, the identification of a graft versus leukemia effect essentially was reported in a couple of papers. One in 1987 by Alex Pfeiffer, where they showed in a large number of patients that if you had a transplant from an identical twin, you had a higher risk of recurrence of disease than if you had a transplant from a sibling. And this is because there is no genetic disparity with an identical twin and therefore no graft versus leukemia effect. So there's an appreciable difference between the two. And then shortly afterwards, Keith Sullivan et al. Described that if you had an allogeneic transplant and you got graft versus host disease and you survived it, that you had a lower risk of relapse of your underlying disease than if you had no form of graft versus host disease again. So implying that there is two sides to the coin, that although graft versus host disease can be bad for you, that of course if you survive it, then you have a higher chance of cure of your underlying malignancy. And then it was, of course, this was refined further so that the major player appeared to be the T cell. And this was very elegantly shown in this registry paper, 1990, and a few other papers at the same time, showing that if you had a marrow graft where the T cells had been removed, and there was a big fad for doing this back in the 1970s and 1980s, because that meant that you didn't get graft versus host disease. However, you had basically the same risk of relapse as if you had an identical twin graft, and the other effects I've noted before. So this means that T cells are very important for fighting off relapse of disease. And this concept was taken forward by a few people about the same time, 1988 to 1990. This being one of the papers. Shimon Slavin in Jerusalem was another such person involved in these things, that giving donor leukocytes, which are mostly T cells, this could be used as treatment for recurrent disease after a marrow transplant, in particular in chronic myeloid leukemia. So the next concept that comes up is the use of hematopoietic cells other than bone marrow. So it was noted a long way back, 1962, that there were hematopoietic cells that circulated in the peripheral blood of mice. And again, this was then proven in dogs, so that you could use peripheral blood cells instead of marrow in dogs. So these are just cells taken out of the circulation of an unperturbed dogs. In this case, they gave the cells after freezing them, which I guess is another important step. And then a number of groups in the 1980s described the ability to mobilise hematopoietic stem cells from patients. So in other words, you could improve the yield of functional hematopoietic stem cells by doing things to the patient or to a donor, to a normal donor. And one of the earliest contributions was this paper published in 1984, British Journal of Haematology, from an Australian group, from Chris Jutner and Lewin Bicteau in Adelaide. And I remember very vividly going to a meeting in Adelaide in 1982, a Transplant Society meeting, and going to Chris's lab and being shown an agar plate taken from a patient with AML recovering from the effects of chemotherapy. And you didn't need a microscope to see the huge number of colonies crowding this agar plate. So these are all hematopoietic cells circulating in the blood from these AML patients. Subsequently, it was shown that you could also do this by perturbing the marrow with growth factors. In this case, granulocyte macrophage colony stimulating factor, or GM-CSF, but it was also shown by the Melbourne group that you could do this with granulocyte colony stimulating factor, GCSF, and get the same sort of results. And in fact, that's really what's proven to be the dominant method these days. So this is showing that you could induce large numbers of hematopoietic stem cells into the peripheral blood in man by giving a growth factor. The final alternative source of hematopoietic stem cells, of course, is umbilical cord blood. And this has an interesting history. So you can see all the authors on this particular paper, but the second one, Hal Broxmeyer, a scientist rather than a clinician, actually floated this idea about 1980 and did a lot of preclinical work showing that this was potentially feasible. However, the preclinical work wasn't actually published until after this paper was published in the New England Journal, essentially proving that you could do this in man. So this is a patient with Fanconi anemia given an umbilical cord blood transplant from an HLA identical sibling, showing that this was a feasible possibility. And subsequently, the use of unrelated cord blood as a source of stem cells has become an industry in itself. Then what do you need to do to the recipient of a transplant? So this wonderful term, euphemistic term, conditioning therapy, was invented back in the Dark Ages sometime to describe how you needed to prepare the patient. Of course, initially, some of the initial transplants done for aplastic anemia, they didn't use conditioning therapy. And these were transplants using identical twin donors for patients with aplastic anemia, where they just gave the bone marrow cells from the donor. And I don't know why these transplants actually worked, because the idea was that the marrow cells were killed and you just replaced them, whereas now we know that most cases of aplastic anemia are actually an autoimmune disease, and you need to do something to switch that process off. So this is a very early summary of the situation with leukemia patients. And you can see here all sorts of different types of approaches to preparing the patients, in other words, giving them anti-leukemic therapy, and plus reducing the host versus graft reaction. And there were very few survivors out of this. However, the Seattle group, around about the same time, reported that if you used HLA identical siblings, you gave whole body radiation plus or minus cyclophosphamide, that you could get long-term survivors. And this was subsequently taken into patients with earlier stage disease, as mentioned before. An alternative is just to use chemotherapy and dispense with radiotherapy. So the Hopkins group, led by Peter Tuchka and George Santos, pioneered the way using busulfan. And this was reported back in the mid-1970s that you could give cyclophosphamide and busulfan, which would allow engraftment of marrow cells in animals, as opposed to giving radiation. And this then led to this being adopted in clinical medicine, such that the busulfan cyclophosphamide regimen is now very widely used. The next concept was, do you really need to give marrow ablative doses of conditioning therapy? So the ideas behind this date back a long way, but essentially this was worked up in the 1980s by Rainer Staub in Seattle. So in this case, they just gave low-dose total body irradiation, a non-lethal dose of radiation, and DLA identical bone marrow grafts to dogs, and showed that you could get long-term engraftment with the appropriate settings. And following that, a number of groups, including the MD Anderson, then used radiation-free regimens, particularly based on fludarabine, the so-called mini transplants or non-myeloablative transplants, where the graft versus leukemia effect was relied on to do the job of curing patients of leukemia without myeloablative conditioning therapy. And the final concept that I want to mention is the one of using histo-incompatible donors. So this has a long history, but the initial successful report goes back to 1998. Of course, there were clinical groups, including the Marsden, and I remember St Vincent's also trying this, using haploidentical donors. But the group from Perugia were the pioneers in actually getting this to work, using a complex regimen of T-cell depletion, high doses of stem cells, high doses of radiation therapy, and a mismatched donor, family donor. The process was simplified by the Hopkins group, who did pioneering preclinical work, experimental animals, relying on the effects of low-dose conditioning therapy, and critically using post-transplant cyclophosphamide as an immunosuppressive agent after the transplant to reduce the risk of graft rejection and also graft versus host disease. And a lot of work was carried out in experimental animals to show that this could actually work, and this culminated in cooperative studies. This is one of the early ones between Seattle and Hopkins, showing that this was a workable regimen in clinical transplantation. So in effect, this is using a 70-year-old drug in a new context to carry out a very important function. So this is a summary. I could have dispensed with all of what I said earlier and just presented this slide actually. But this just summarises what I said, that the first attempts to treat acute leukemia with bone marrow transplantation go back to the mid-1950s. Transplants were also applied for patients with, who were radiation accident victims, again without any knowledge of histocompatibility. First reports of successful transplants using identical twins for leukemia in the late 1960s, as were transplants for severe combined immunodeficiency. And then successful transplants reported for early stage leukemia in the late 1970s, and unrelated donor transplants first successfully carried out in 1980, and then 1988 the first successful umbilical cord blood transplant. So what about on this half of the world? I can remember going to the operating suite in the emergency department in 1974 with the entire haematology department run by Harry Cronenberg in order to carry out a bone marrow harvest. There were about 40 people in the operating suite, which was a pretty tight fit. And the whole thing was supervised by a guy called Len Johnson, who was an Australian who'd worked in Seattle up until just before then, who had a job down at the Children's Hospital. And he patiently took Harry and the rest of us through the process of a bone marrow harvest and then how to treat a patient, a young guy with aplastic anemia who was in Harry's care. And this was a big deal. I remember after each dose of cyclophosphamide, everyone gathered around the ECG machine to see whether there was cardiac damage from giving the high doses of cyclo, things that we just wouldn't even bother thinking about these days. And unfortunately, this patient died, and when I thought back on it later, there were so many things that we didn't know, and there was no cyclosporin. You know, it was a very early adventure. As I remember, and Roger Skerm will probably correct me on this, Jim Biggs and colleagues at St Vincent's did a successful transplant prior to 1974. Is that right, Roger? Sorry? 76, was it? Okay, I thought James was the first one to do one. Anyway, I'm wrong. Sorry? That survived. Okay. Okay. I'm not sure about paediatric centres. Anyway, then progressively more and more centres opened up. Westmead opened up in 81. and North Shore, and then Max Whiteside in Melbourne did some transplants for aplastic anemia, I think around 1980, and then sent his registrar, Jeff Scher, over to Seattle to learn how to do it properly, and so on and so forth. I guess the question, you know, this is essentially technology transfer, but, you know, what role did we really play in the grand scheme of things? Were we just, you know, foot soldiers? But I think there were two really big contributions that Australians made to the field, and this is being very selective, and this is going back a while. One was the work done at the Weehigh and the Royal Melbourne by Dick Fox and George Mawson, Bill Sheridan, and so forth, with granulocyte colony stimulating factor, a whole series of papers showing that this, as I said before, stimulated the mobilisation of cells into the blood, and these could be used for transplantation, or they could be used afterwards to stimulate marrow regrowth. So this was a very important contribution. I think the other very important contribution was from Chris Juttner and Vic Toe in showing that you could do autografts using the cells harvested from patients after recovery from chemotherapy. And then, of course, the two were put together, which is now essentially standard practice in marrow transplantation. So that's where history ends. The future, you can see very clearly here, the way forward. I'll leave it at that. I think people will probably be more interested in lunch at this stage as the way forward. I'm happy to answer any questions, otherwise discuss this after lunch. I'll take questions, but no criticism.
Do you mind if I ask a question then? Out of all the things that you presented today, what do you think has been the most important change in knowledge that's reflected an improvement in outcome over the last 10 years or so?
I don't know, how many pharmaceutical reps are in here? Should have got them to declare their conflicts of interest. It's impossible to say. Obviously, I guess in the last 10 years of my career, it was just amazing to see the introduction of successful haploidentical transplantation. Because for so long we were focused on, you know, finding the donor. Initially, I can remember up into the end of the 1980s, it was a lottery. You know, you either had a matched sibling or you didn't. If you didn't have a matched sibling, there's the exit, and you know you weren't eligible. And then the introduction of matched unrelated donors came in, which changed the picture, but not completely. Although, you know, now I think in places like Germany, you know, if you're a German, you can find a donor from the German registry for just about everybody. Well, that's what Nicola Gockberget said the other day. Yeah. But I think you know, in this society, being able to find a suitable donor that will work for just about every patient is a big step forward.
You mentioned that doing randomised trials and transplantation is very good. Really been a good experience, I suppose, over the last few years. Can you perceive exactly what barriers there are to performing a randomised trial in allogeneic transplantation, and how likely you have to overcome those in the future?
I think the most important thing for a randomised trial is to know in advance what the answer will be. And if it's going to be a marginal difference, then just forget about it. But because you could see with ours, you know, with Seattle, that, you know, they had very modest numbers, but they picked the right question. Yeah. That's evading the point. Sorry.
No. All right. Okay.
I was fortunate to go a week ago to a talk by a fellow in the audience here, David Gottlieb, who could have answered that question very succinctly. But really the last remaining obstacle, major obstacle, is the prevention of recurrent disease after a transplant, because despite all these advances, that problem still hasn't been solved. It works for some people, but not for other people, unfortunately. And the reasons for that still aren't properly understood, and the way to get around that still isn't fully known. But at least we have lots of hints from what I've mentioned here about the power of the immune system in some people to be able to cure malignancy. And it's about discovering why it works for some people but not for other people that's important. And there are ways forward in trying to improve the way the immune system responds to malignancy after a transplant. So there's genuine cause for hope, I think.